All human cells come originally from single cell. The cell divide, giving rise to two cells, and then each of the new cells divide again, and we get four cells, and so on. After some time, the new cells start to differentiate. So some cells become specialized in secreting hormones, and others in contraction, and others in absorption. Cell division and differentiation are hallmark of human, and they are controlled by proteins and genes. Cell cycle represents the start of one cell to get new cells. It is a cycle that is repeated again and again to yield new cells. During embryonic life and early childhood, this is essential for growth. As we get older, cells become more specialized, and sometimes we have types of cells that require to stay still and no more division. Cells become categorized in three types: permanent cells that do not divide anymore, like neurons; stable cells that only divide in special circumstances, like liver cells; and cells that are continuously dividing, for example, epithelium and bone marrow cells. From the birth of a new cell till cell division, the cell passes into different stages: growth 1 and 2, named G1 and G2, synthesis phase or S phase, and mitosis or M phase. These phases represent the steps of cell cycle. Cells that stop dividing, like permanent cells, enter a phase known as G0 phase immediately after division. During S phase, the genetic material of the cell is duplicated. G1 phase prepares the cell for that and increase organelles and cytoskeleton elements. In G2 phase, proteins needed for cell division are prepared and move to their proper sites. Why we mention and need to know all this story? Well, because cell cycle is tightly regulated. Neoplasia means new growth, and cancer develops if we have cell division that is uncontrolled and not needed. Suppose we get defective DNA. Is it wise to go ahead and synthesize this DNA? The answer is no. And we don't want the cell to enter S phase in that case, because if we have new defected genes produced and passed to the next generation, tumor can result. Thus, Allah subhanahu wa ta'ala created checkpoints between steps of cell cycle. If a person have money in one country and want to pass via 3 other countries to have business and then come back to his original country to multiply that money, we are going to have checks for his money, amount, reality, etc. at each airport of a new country. This is what happens in cell cycle. Cyclins are proteins working in cell cycle as passport officer. An example of these proteins is the retinoblastoma protein. This protein is a regulator for G1 S phase transition. It can be in active or dephosphorylated status, in which it doesn't allow cell to enter the S phase, or it can be inactive or phosphorylated state, in which the cell is allowed to pass from G1 to S phase. It is similar to harsh passport officer. When present in his uniform, does not allow anyone to pass, and when he is home and relaxed, everybody is able to enter. When the cell is stimulated by growth factors like cyclin D, retinoblastoma protein is phosphorylated, so become inactive, and in terminally differentiated cells, retinoblastoma protein stays dephosphorylated and inhibit transcription. An 18-month child, we examine her eye and find white pupillary reflex, something called leukocoria. When biopsy is taken from that lesion, it is found to be a cancer of her retinal tissue. Retina is the last layer in the eye. It is the neural tissues that is responsible for vision. This child lost her vision in one eye due to the cancer, and she suffered side effects of chemotherapy needed for treatment. The basic cause of this child problem was defect in retinoblastoma gene. The results of this defective gene is retinoblastoma protein that is defected and lack function. Cell cycle in the eye retina continues unchecked, and the retinal cells divide in uncontrolled fashion, resulting in tumor. Genes are codes for protein synthesis. If a gene is defected, the resultant protein is defected. In our cells, there are genes that code for proteins that prevent the development of cancer, similar to retinoblastoma gene. These genes are known as tumor suppressor genes. Normally, the proteins of these genes prevent progression of cell cycle. If there is defect, cell cycle continues and tumor develops. There are other types of genes involved in tumor pathogenesis. For example, oncogenes, opposite to tumor suppressor genes, are normally inactive. When they become active by mutation, there results in uncontrolled growth. Also, there are genes that regulate or prevent apoptosis, which is programmed cell death. Overexpression of these genes is associated with cancer, and factors to inactivate them are researched to be method for cancer treatment.